Welcome to Aquatic Biology. Have you ever wondered how scientists keep track of millions of fish in the vast, seemingly endless ocean? Today, we are going to explore the fascinating divide between the open ocean swimmers and the ocean floor dwellers. We will learn not just what these marine resources are, but exactly how we use mathematics to measure, monitor, and protect them for future generations. Let us dive right in. To begin, imagine looking at a massive, multi-story glass aquarium. The fish swimming freely in the middle and upper levels, never touching the bottom, represent our pelagic resources. The word pelagic comes from Greek, meaning open sea. These are the marathon runners of the ocean, built with highly streamlined bodies for continuous swimming across thousands of miles. They live in the vast water column, from the sunlit epipelagic zone down to the twilight mesopelagic zone. Because they live in open water, they rely heavily on ocean currents and the primary production of microscopic phytoplankton, which forms the absolute base of their food web. Now that we understand what pelagic resources are, let us look at why they matter so much. These open ocean swimmers are absolutely vital to both global food security and the health of marine ecosystems. Small pelagic fish act as a crucial energy bridge. They consume microscopic plankton and, in turn, become food for larger predators, marine mammals, and seabirds. Commercially, they make up a massive percentage of the world's total fish catch. They provide an affordable source of protein for billions of people and are harvested to produce essential fish meal and fish oil. Without healthy pelagic populations, the entire oceanic food web would simply collapse. Because these pelagic fish are constantly moving across vast oceans, scientists can't simply count them by looking at the water. This brings us to a vital mathematical metric called catch per unit effort, or CPUE. Conceptually, this formula acts as a proxy for population size. In the equation, C represents the total catch, usually measured in kilograms or metric tons. E represents the effort expended to catch those fish, like the number of hooks used or hours spent fishing. If the catch C increases while the effort E stays the same, the CPUE goes up, indicating an abundant, healthy population. Conversely, if fishermen must increase their effort just to maintain the same catch, the CPUE drops, warning us of overfishing. Before we move on, let us see this formula in action with a concrete example. Imagine a commercial fishing fleet targeting yellowfin tuna. Over 1 month, we are given that they catch a total of 45,000 kilograms of tuna. To achieve this, they deployed 1,500 fishing hooks. We are asked to calculate the catch per unit effort to understand the tuna's relative abundance. We choose the CPUE formula to standardize our data, removing the bias of simply looking at the total catch. We substitute our values, dividing 45,000 by 1,500. This gives us a final answer of 30 kilograms per hook. Physically, this means every single hook deployed yields 30 kilograms of tuna, providing a baseline to monitor the population's health next year. Having explored the open ocean, let us now dive down to the ocean floor. If pelagic fish are the birds flying in the open sky, demersal fish are the creatures living on the forest floor. The word demersal comes from a Latin word meaning to sink. These resources consist of fish and invertebrates that live and feed on or near the bottom of the sea, occupying the benthic zones. Unlike their pelagic cousins, these animals do not need to swim constantly. Instead, species like flounders, lobsters, and crabs are adapted to resting on the bottom, camouflaging against the dirt, or burying themselves in the sand to ambush prey. Their entire life cycle is intimately tied to the physical structure of the seabed. Now, why do these bottom dwellers require our special attention? Demersal resources are incredibly important because they represent some of the highest value seafood products in the global market. Species like lobsters and scallops command premium prices and support millions of coastal livelihoods. Ecologically, they play a critical role in nutrient cycling. They scavenge dead organic matter that sinks from the upper layers, preventing the seabed from becoming a toxic wasteland. Furthermore, because they are relatively stationary compared to pelagic fish, they are excellent indicators of localized environmental health. If a specific seabed area becomes polluted, these demersal resources will be the very first to show signs of decline. Because demersal fish live deep on the ocean bottom, we cannot track them with standard sonar. To estimate their total biomass, scientists use the swept area method. Conceptually, this involves dragging a net over a known area and mathematically reconstructing the true population based on what escapes. In this formula, B is the total estimated biomass. C is the actual catch weight brought up. The variable A represents the physical swept area the net dragged across. Finally, X is the catchability coefficient, representing the percentage of fish actually caught. If the catchability X is very low, meaning most fish escape, the formula mathematically compensates by increasing the estimated total biomass B to account for the ones that got away. Let us apply this formula to a real-world survey. We are given that a research vessel drags a net over a swept area of 2 square kilometers. They bring up a catch of 500 kilograms of flatfish. We also know the net only catches 25% of the fish in its path, so our catchability coefficient is 0.25. We are asked to estimate the total biomass. We choose the swept area formula to ensure we do not underestimate the population just because our net is imperfect. We substitute our values, multiplying 2 by 0.25 in the denominator to get 0.5. Dividing 500 by 0.5 gives us a final answer of 1,000 kilograms. Physically, this tells us that the actual amount of fish living on that seabed is twice what we caught. Now that we have used this formula, let us understand the mathematical reasoning behind why it works. What are we actually doing when we multiply the physical area by the catchability coefficient in the denominator? We start with the physical area swept by the net, which is A. But we must recognize that our gear is imperfect. It only catches a fraction of the fish, represented by X. By multiplying A and X, we are calculating the effective area sampled. In our example, multiplying 2 square kilometers by 0.25 gives us an effective area of 0.5 square kilometers. The intuition here is that we are essentially shrinking the physical area to represent a smaller, hypothetical space where we caught absolutely everything. Continuing from our effective area calculation, let us see how this leads to the final result. We take our actual catch, C, and divide it by the effective area, which is A times X. Because our catchability coefficient is a fraction less than 1, dividing our catch by this smaller effective area will always result in a larger number. This makes perfect logical sense. We know we missed fish, so the true biomass must be significantly larger than our actual catch. This mathematical trick perfectly compensates for the fish that got away, allowing biologists to reconstruct the true population size hidden in the dark depths and set sustainable fishing quotas. So, we have proven how to measure these populations. This brings us to a visual summary of everything we have discussed. Look at this cross-section of the ocean. At the top, in the light blue water column, we see the pelagic zone. Notice the streamlined sardines and tuna swimming freely. The fishing boat is dragging a mid-water net through this zone to sample the pelagic resources. Now, look down at the sandy seabed, labeled the demersal or benthic zone. Here, you can see flatfish and crabs resting directly on the bottom. To sample these creatures, the boat uses a completely different method, dragging a bottom troll net right across the sand. This diagram perfectly illustrates the fundamental divide in marine habitats and the different gear required to study them. To wrap up our lecture, let us review the most important ideas. First, we learned that pelagic resources are the fast-swimming, highly migratory species of the open water column that drive the global food web. Second, in contrast, demersal resources are the bottom-dwelling species, intimately tied to the seabed, offering high economic value. Finally, we saw how mathematics is essential to marine biology. By using tools like catch per unit effort and the swept area method, scientists can monitor both of these distinct groups. You now understand the fundamental divide in marine resources and the tools we use to ensure our oceans remain full of life for generations to come.